right, Keith, thanks so much for that introduction. And I'm really excited to be here today. I've really enjoyed doing this science, and I hope that you enjoy hearing about it. It's been a fun 5.5 years, pretty much, since I've been working on this with Alex. I have no disclosures. So we're going to wander through a few different topics today. So let me just give a quick roadmap of where we're going to be going. So I'll start by reviewing some of the descriptive epidemiology and clinical features of syphilis that Dr. Lorenzo Giacchini touched on during his grand rounds last year. Then we're going to focus on how Treponema pallidum causes disease, and I'll tell you about the microbiology and immunology of syphilis and how genomic studies have really helped to shed light on how this stealthy pathogen operates. I'll also highlight some recent genomics findings, including global population structures and national surveillance. And then finally, we'll take a deep dive into a single patient sample and discuss what it tells us about T. pallidum evolution. So syphilis is a sexually transmitted bacterial infection, as you know, caused by Treponema pallidum subspecies pallidum. Globally, syphilis remains very common with over 8 million incident cases per year. Syphilis can also be vertically transmitted from a pregnant woman to her fetus, and congenital syphilis causes more than 220,000 fetal and infant losses each year, and the highest burden is in sub-Saharan Africa. And I think the most vexing thing about syphilis is that we can treat it. Penicillin still works great, even after 80 years, and we still have this enormous burden. And so a lot of the goal of the research right now is to develop a vaccine, and that's the context of a lot of the investment in genomics that I'm going to be telling you about. In the United States, we're really at an inflection point in the syphilis epidemic. More than 209,000 cases were reported in 2023, which is the highest since penicillin was widely available. During and after the AIDS epidemic, or the worst of the AIDS epidemic, syphilis became a national public health priority, and in the year 2000, there were fewer than 6,000 cases. And then what happened? We took our eye off the ball. We haven't been prioritizing syphilis and STIs, and health departments are cash-strapped, as we know. There's also been the role of behavioral changes. So now that PrEP is more widely used to prevent HIV infection, there's more unprotected sex and more transmission of non-HIV STIs. And then finally, we've seen persistent global shortages of injectable long-acting penicillin and had to resort to second-line antibiotics, although I think that is more or less resolved at this point. So a big change in this phase of the epidemic, though, is who is getting syphilis. In the last 10 years, we've seen an explosion of cases among women, and when more women of childbearing age are getting syphilis, the rates of congenital syphilis increase. In 2023, in the US, there were almost 3,900 cases of syphilis, which is the most on record since the early 90s. And importantly, about 90% of those cases can be attributed to a lack of testing or treatment or insufficient treatment, and a lot of these women just didn't have prenatal care. So these women and babies have just really slipped through the cracks. And there's a huge difference in rates of congenital syphilis among different demographic groups. But the situation among Indigenous people is really particularly problematic. Fully 3% of Native American babies born in South Dakota in 2023 had congenital syphilis. So this is not a situation that's going to improve without investing heavily in public health infrastructure in these communities. And I think the concentration of syphilis among certain demographic groups is hardly news, and syphilis, and especially congenital syphilis, is really associated with adverse social determinants of health. These disparities don't arise from nowhere, though, and I think now is a really good moment to remember the very uncomfortable history of the role of the United States Public Health Service in syphilis research. Understandably, it sowed deep mistrust of the medical establishment among some marginalized communities, particularly communities of color, and that's a very important barrier to accessing healthcare. So the most infamous of the PHS studies was the Tuskegee study of untreated syphilis. Starting in 1932, the USPHS coerced participation from hundreds of Black men with syphilis in rural Alabama, and they told them that they would be examined and treated for their bad blood. The reality was that doctors wanted to observe the long-term effects of untreated syphilis, especially upon autopsy. These men were then denied treatment even when penicillin became widely available. Many men died from advanced syphilis, transmitted it to partners, and several babies were born with congenital syphilis. In a separate study in Guatemala in the late 1940s, prisoners and mental hospital patients were deliberately infected with syphilis to test new treatments, like a new form of penicillin. The PHS also infected prisoner volunteers in Sing Sing prison in New York. And one thing I want to mention is that far from being conducted in secret, these are studies that were approved at the highest levels of government, including final sign-off from the Surgeon General. And so when the existence of the Tuskegee study finally became public in 1972, it really, I think, forced a reckoning of how we conduct medical research in this country. In 1974, the National Research Act was passed, which mandated the creation of institutional review boards, IRBs, to oversee human subjects research. And it commissioned the Belmont Report, which really boiled down the guidance for those of us doing human subjects research to 3 core principles, which are simply respect for persons, beneficence, and justice. To fully understand how deeply unethical and cruel the Tuskegee study was, I'd like to take just a few minutes to discuss the progression of untreated syphilis. Following exposure to T. pallidum during sexual contact, a few weeks will generally go by before the first symptoms of infection occur. At that primary stage, classically, there will be a painless lesion called a chancre that forms at the site of infection. The chancre will ultimately heal on its own, but not before bacteria disseminate. And those can then come back and recur as one of several manifestations of secondary syphilis, most commonly a rash. After resolution of the secondary disease, syphilis will often become latent for years or even forever. In about 30% of people, the disease will progress to an unusual array of tertiary disease presentations, including cardiovascular damage, aortic aneurysm. There's lots of different bone and skin damages and some different kinds of neurosyphilis. And for this reason, it's been called the Great Masquerader. And scientists have been studying Treponema pallidum for over 100 years because it can cause such an unusual array of symptoms. It seems almost limitless. It's difficult to study, though. It needs host cells, and so we can't study it in isolation. Until a few years ago, that meant that it had to be passaged in rabbits, and now we're very fortunate to have an epithelial tissue co-culture system, or an epithelial cell co-culture system. And to make matters worse, there wasn't a genetic system at all until 2021, and we still don't have a good system to do mutagenesis screens to understand gene function. So a lot of what we know are from biochemistry and serology studies, and of course, clinical studies. I think some of the most critical findings from those, if you can summarize just to a few, are that you need both antibodies and Th1 T cells making interferon gamma and activating macrophages to eliminate treponemes. We also know that the right vaccination scheme with enough doses can achieve protection. And finally, T. pallidum has a really weird outer membrane, and we'll talk about all of these a bit. And what I want to do now is try to explain some of these observations through the lens of T. pallidum genome features. So Treponema pallidum was one of the first bacteria to have its genome sequenced, and some of the methods that were used will seem familiar today. It was early library preparation, shotgun library, and de novo assembly that we now consider routine, but were cutting edge at the time. And the one thing that I really want to impress here is that the protocol for extraction of pure treponemal DNA was absolutely punishing. Multiple rabbits, at least 4 ultracentrifugation steps, southern blotting to confirm purity. Just an incredible amount of effort to get about the same amount of bacteria, same amount of genetic material as an overnight culture of E. coli. So where did this heroic effort get us? So I'd like to introduce the Nichols strain of Treponema pallidum, which was originally isolated in Washington, D.C. in 1912 from a patient with neurosyphilis. And it's been propagated continuously in rabbits since then. And it's by far the most widely used strain in the laboratory. The Nichols strain genome is very minimalist. All T. pallidum genomes are very minimalist at just over 1.1 megabases with no mobile genetic elements. It's chucked a lot of metabolic pathways, and because of that restricted metabolism, it's really dependent on its host for nutrients, and that's why it can't be cultured in isolation. T. pallidum is a stealth pathogen that needs to be able to disseminate undetected. And to do that, it has a very immunologically quiet outer membrane. So it doesn't have LPS, and it doesn't have very many outer membrane proteins that our immune systems will innately recognize. And an added challenge is that it's very distantly related to some of the more well-studied gram-negative bacteria. And a lot of the predicted proteins in Treponema pallidum have no known orthologs, so we don't know what they do. And this includes a family of 12 of the 12 T. pallidum repeat, or TPR paralogs, which comprise almost half of the known outer membrane proteins. So now that we know a bit about the genome, let's add a little bit more info here. So first, there are 2 clades of T. pallidum subspecies pallidum. These are named after the Nichols and SS14 strains. And for the rest of the presentation, if I refer to Nichols and SS14, I will mean the clade and not the strain. I'm sorry, Sheila. In addition to T. pallidum subspecies pallidum, which causes syphilis, T. pallidum subspecies Pertenui and Endemicum cause Yaws and Bajil, respectively. And those are also progressive chronic bacterial infections that primarily affect poor children in the developing world. Pertenui and Endemicum aren't considered to be sexually transmitted and aren't considered to be as invasive. They don't seem to cause congenital infections. But that's fascinating, and I want to emphasize how very similar these strains really are to each other. There's less than 0.1% of the core genome that's different between Endemicum or Pertenui and pallidum. And between the SS14 and Nichols clades, there's only about 120 SNPs different in the core genome. So that's very, very low diversity, and I think really speaks to T. pallidum's success as a pathogen. It's clearly under some pressure to hold its genetic shape. In contrast to the lack of diversity seen in the core genome, there's one single locus, TPRK, which is one of those TPR proteins I mentioned earlier. And TPRK is highly heterogeneous within a single sample. And I really love talking about TPRK because UW has been just really leading the way in our understanding of this gene for over 25 years. The TPRK gene has 7 variable regions, and thanks to AlphaFold, we can now predict TPRK to be an outer membrane porin, a 20-stranded beta barrel with variable regions corresponding to 7 host-facing extracellular loops. And those are constantly evolving so that they can stay invisible to the immune system. And then it evolves, new variants take over as the adaptive immune response ramps up against the prior. And this is a pretty common evolutionary strategy for STIs. And the way that T. pallidum accomplishes this is to use non-reciprocal gene conversion to swap out some sequence fragments of the TPRK variable regions. So these sequences come from about 50 donor sites that are on the same chromosome. Each one of them is specific for one of the TPRK variable regions, and I have the colors coordinated there. So there's these sites of microhomology within the donor and acceptor sites that then allow homologous recombination to create new chimeric sequences at the expression site. And this might swap out 1 or 30 nucleotides or anything in between. And these new variants then accumulate during infection and can escape immune elimination. To illustrate what that looks like in human samples, here's a phylogeny of 629 full-length TPRK protein sequences. And those were recovered from a cohort of just 13 patients. Each patient is a different color, and each tip is a unique sequence. None of the sequences showed up in more than one patient. And what I really want to show you here is that there's just an incredible amount of diversity that can be generated in TPRK. Essentially, we think of it as being on par with the number of unique antibody sequences that can be generated through VDJ recombination. To more formally show the role of antigenic variation in immune escape, our collaborator, Dr. Lorenzo Giacconi, generated a strain in which the donor cassettes had been replaced with a kanamycin resistance cassette. And that should knock out antigenic variation. And then he infected rabbits intradermally at 10 locations on the back. And as you can see on the right panel there, the animals infected with the wild type strain, the bacteria escape immune clearance, and that results in growth of these really nasty ulcerated lesions by day 72. When the animals were infected with the knockout strain, the lesions were cleared without any treatment. And when we actually looked at the genomics of TPRK in these animals, we saw something, this made a lot of sense. And I'm going to show a really complicated figure here, so bear with me while I set it up. In the left panel, I'm going to show wild type results, and on the right is going to be the knockout. Across the top of each panel, or across each panel, are going to be columns, and each of those will correspond to a single biopsy sample. On the y-axis, I know you can't read the text, but those are unique amino acid sequences of TPRK variable region 5, which is the one that we're looking at here, that we found when we sequenced the lesions. And importantly, only the sequences that we didn't find in the inoculum. So these are unique ones that were formed during the infection. And so these colored tiles then represent the proportion of total TPRK V5 each of these amino acid sequences takes up in that particular lesion biopsy sample. And you can probably appreciate that new sequences are readily being made in each lesion in the wild type strain, and these allow escape of the adaptive immune response. In contrast, the strain that doesn't have these donor sites, we don't see any new TPRK sequences being generated, and the adaptive immune response cannot eliminate the treponemes. So let's dig in a little bit more and look at some of the most current insights from whole genome sequencing studies. So if we're going to make a vaccine, we need to know about the rest of the outer membrane proteins that don't undergo antigenic variation. But this presents a challenge. Remember, we can't streak T. pallidum on a plate and sequence a colony. So we have to find ways to enrich treponemal DNA. Otherwise, you just end up sequencing host DNA. And those previous heroic sequencing efforts that involved a lot of animals and ultracentrifugation, this is just not going to work if you want to study hundreds of samples. So the solution to this is to use a technique called hybrid capture sequencing. And in this technique, we have a library of thousands of oligonucleotide baits that tile across the T. pallidum genome and are complementary to it. These baits are then added to the fragmented sample DNA, and the DNA fragments that are complementary to the baits are bound. So that's going to be the T. pallidum DNA should specifically bind the baits. They're captured, and then they can be magnetically separated from unbound host DNA. And after some very careful washing, those target sequences have been enriched in our hands by about 10 to 100,000 fold. And this is actually how we started studying syphilis. Alex had a ton of experience using hybrid capture for HSV and was included as the genomic score for a successful U19 application for syphilis vaccine development. And that was a grant led by Dr. Anna Wald. And so thanks to the development of hybrid capture, and just as importantly, I think, more importantly, investment by major funding organizations to help develop a vaccine like that U19, the last several years have really seen an explosion in whole genome sequencing. So I'm not gonna go through every study. I'm just gonna summarize a few of these findings. So first, the SS14 clade really took over starting in the 90s after a population bottleneck. And you can see how shallow some of these branches are in the SS14 lineage, which is in red there. And that indicates that the strains are more similar and probably haven't had as long to diversify as the Nichols lineage. And in contrast to this core genome, these studies of syphilis samples worldwide, we can now appreciate more fully how diverse the handful of host-facing antigens are. And that has really important implications for vaccine design. We also learned a bit about antibiotic susceptibility. So although there have been some SNPs in a few of the T. pallidum penicillin binding proteins, none of them seem to affect beta-lactam susceptibility. We also looked at the distribution of a well-known SNP in the gene encoding the 23S ribosomal RNA, and that confers resistance to macrolide antibiotics like azithromycin. And we also confirmed that macrolide resistance is much more prevalent in high-income countries. The last thing I'm going to mention, I think, is really interesting. So you remember how I told you that there are 3 subspecies of T. pallidum and only subspecies pallidum is sexually transmitted? Well, it's almost certainly more complicated than that. In a cohort of Japanese men with syphilis, when we did the sequencing, it turned out that several were actually infected with T. pallidum subspecies endemicum, which is clinically indistinguishable from subspecies pallidum. And this had been seen in other samples from Japan and has also been seen in rare samples from Cuba and I think from France. And there's clearly, I think, more of a continuum than we had initially appreciated. And we're going to see how that continues to play out as we fill in those evolutionary branches a little bit more. So all of this sequencing is great, but it does present a bit of a problem. As these samples were rapidly added to the Sequence Read Archive, or the SRA, which is the public database for genomics data, all of the groups were using different hierarchical clustering schemes for their data analysis. And I've put the figure legends up here for a number of papers that I refer to regularly. And you can see none of these have the same clusters in them. And what this means is that all of these papers are good, they're valid, their conclusions are strong, but we can't compare them. There's just not really a good way to look at the worldwide distribution of strains when we don't have a constant cluster scheme. And so another challenge to the state of the data is that the vast majority of T. pallidum data on the SRA hasn't been processed into consensus sequences. The consensus sequences are the ones that you want because they're on GenBank and they're searchable in BLAST. And so unless you have considerable genomics expertise, a lot of the T. pallidum sequencing data remains more or less inaccessible. So to address both the disorganization, I think, and challenges of data accessibility, we're really, I think, fortunate that the Gates Foundation saw some of our streamlining proposals to be an important extension to what they were already investing in the genomic space. And so we just got started a few months ago alongside colleagues at the Sanger Institute and Tübingen University. And we really have three main goals here. So first, we're going to develop that clustering scheme. And that can be hopefully adopted by everyone in the field so that we can talk to each other a little better. We're going to make some consensus sequences and make those available. And finally, we're going to add all of this information to a customized NextStrain instance for T. pallidum. So for those of you that aren't familiar, NextStrain is an interactive platform for genomics data exploration. And you almost certainly saw screenshots of it if you didn't use it yourself during the COVID pandemic. And we think it's a really optimal tool for exploratory analyses and understanding the global distribution of vaccine targets. And I think really good for hypothesis generation. So we're just a couple of months into that project. And the first thing that we did was to download all of the raw data for more than 3,000 strains and process them using a new simplified assembly pipeline. And here I need to give major credit to Jeff Furlong and Alex's group for getting that pipeline up and running just really in record time. And so here's where we stand. We now have so many strains that we need to switch to a circular phylogeny. So you can see some of the same trends from earlier. Shallower branching in the SS14 clade than Nichols, consistent with more recent expansion. And there have only ever been a few endemicum samples sequenced, so you might not be able to see them very well, but they're in green at about 3 o'clock there. I think there's 8 of them. And when I color the tips by continent, you can immediately appreciate that there's some geographic clustering in these clades. So at about 2 o'clock in blue, those are all Yaz samples, Treponema samples from a mass drug administration trial in Papua New Guinea. And then at about 11 o'clock in the red, those are mostly samples from Madagascar and Malawi and South Africa. And these are places where we maybe don't think of people as having much geographic mobility. And in contrast, you can see this almost clonal cluster in SS14 with primarily samples from North America, Europe, and Australia. I'll also point out some of these very distant samples with arrows here, including this one from Chile at about 1 o'clock that actually doesn't fit in with any subspecies at all. These are all samples that have been extracted from human remains, from bones at archaeological sites. And the goal of that is to understand treponemal evolutionary history. I'm not going to talk about the Columbian hypothesis today, but a lot of it is to understand where T. pallidum came from and how it was initially introduced to Europe. So take a look at these two samples that I've highlighted in yellow. So one is a sample from Mexico dated to the 1400s, and the other is from Lithuania from probably sometime between the about the 1400s and 1600s. And they are most closely related to a cluster of non-human primate samples from Tanzania and yaws samples from Ghana that were collected between the 80s and now. And so this really speaks, I think, to some of the challenges with T. pallidum genomic epidemiology. The exceptionally slow rate of evolution in the core genome really limits our understanding of transmission. In the United States, largely because we almost always use serology to diagnose syphilis, we're really undersampled in this country, and there's only been about 200 samples ever sequenced. Many of those were collected 20 or more years ago as well, which really limits their utility for current analyses, including for genomically encoded antimicrobial resistance. And now, I've already told you that syphilis continues to remain susceptible to penicillin, but sometimes penicillin isn't an option. And so azithromycin was initially proposed as an alternative treatment for penicillin allergic patients. And that was based on a trial done between 1995 and 1997 that showed that azithromycin, a single dose of azithromycin, had equivalent efficacy to BPG to treat early syphilis. However, the first cases of azithromycin resistance came very quickly after that. And in San Francisco, the prevalence of that predictive 23S mutation went from 0 to 56% in just 4 years. And this was recapitulated in a larger cohort collected between 2007 and 2009. And again, it suggested that about 53% of samples total across all patients were resistant to azithromycin with different enrichment in different demographic groups. And since then, in the limited sampling that we've done, rates of the 23S mutation that confer azithromycin resistance have continued to creep up. And by the time I started working on syphilis about 5 years ago, it was really generally accepted that all syphilis in the United States and other high-income countries was nearly 100% azithromycin resistant. So flash forward to a few years ago, when I think really the perfect storm of a lot more syphilis causing increased demand and also some supply chain pressures meant that there was almost no injectable penicillin available in the United States. Twice-daily doxycycline for 2 weeks is the recommended second-line antibiotic for early syphilis, but in some patients, that can be really difficult to complete. And so we had heard some anecdotes from our clinical colleagues in ID about some clinicians that were giving a single oral dose of azithromycin because they didn't have BPG available. These are just a few anecdotes from around the country. That's not in UW or anything, but they were giving patients the single oral dose of azithromycin because they didn't have BPG available and they knew that their patient would not be able to complete that 2 weeks of doxy. And what we realized is that there was really a need for an unequivocal communication about the current rates of azithromycin resistance. And so we needed as comprehensive a data set as we could get. And thanks to a huge effort involving the CDC and some Canadian public health teams, we really tossed the freezers and scraped together every sample that we could possibly find. And we determined the 23S allele using whatever methods we could. So we used whole genome sequencing, PCR and Sanger sequencing, allele-specific qPCR, restriction fragment analysis, whatever we could. And unsurprisingly, out of the 604 samples collected in 16 states and provinces since 2017, only 5 were sensitive to macrolides. And among samples with whole genome sequencing, we showed that resistance is throughout the entire phylogeny in all lineages. So we were able to deliver a very strong and unequivocal public health message that there's no place for the use of macrolide antibiotics against syphilis in North America. Okay. And for the rest of our time today, I'm going to change it up a bit and do a very deep dive into a really interesting sample that had been through both pathology and molecular micro for a diagnosis, and I guess also immunology as well. And then came to me as a research specimen. And I know that some of our clinical lab staff do a lot of work to put remnant samples together for research. And today I just want to say thank you and share a little bit about what we learned about syphilis from this one patient. So let's set the stage. In support of the azithromycin study that I just showed you, I had gotten a batch of about 20 remnant eluates that had tested positive for Treponema pallidum by a molecular test from my longtime collaborator at Molecular Micro, Dr. Joshua Lieberman. I get very limited metadata for patient samples, but what I did know was that this is a stomach biopsy from a male patient living with HIV. So we did the sequencing, and the T. pallidum sequencing looked great. And that's especially considering this was out of FFPE. It was formalin fixation and paraffin embedding really damages DNA. And so in spite of that, we still had great coverage, very even, very deep. But when I manually inspected the way the reads aligned to the reference genome, here I was surprised to find mixed template at positions that discriminate between the Nichols clade and the SS14 clade. And this had never been reported before. And as anyone that works in a molecular lab knows, when you see mixed template, the first reaction is never, boy, that's interesting. It's more like, oh boy, how did I manage to cross-contaminate this? So obviously resequencing was the next step. So we requested a recut of the stomach biopsy from Northwest Biospecimen. And it turned out that there was a colon biopsy from the same patient as well. And that hadn't been sent for a molecular diagnosis originally. When I talked to Josh a bit more about this case. He pointed out that this was a medically very interesting case, and he'd been involved in writing up an abstract with the GI team, since gastric syphilis is just such an unusual presentation. And so here is what we knew and what we learned while we were waiting for cuts of those FFPE biopsies. The patient had a history of appropriately treated primary syphilis 18 months prior. He was pretty sick by the time he presented to the UW-ED. He'd been living with blurry vision and eye pain for about 4 months, but that had progressed to really having light perception only. He was almost blind. On eye exam, he had dense vitritis in the anterior chamber, and his CSF showed elevated protein in nucleated cells and low glucose. He also had a 6-month history of GI distress, and that had led to about a 40-pound weight loss. So the GI team started with endoscopy to see what was going on. I am not a physician, but I'm reasonably confident that there should not be that much hemorrhage or pus in the stomach. And when the biopsy tissue was sent to pathology, Dr. Brian Mao, who was a fellow at the time, and Dr. Paul Swanson found infiltration by neutrophils and plasma cells on H&E stain. And in panel D here, a stain for T. pallidum was positive, sometimes with these wispy little spirochete forms inside neutrophils. And if that seems subtle, that's part of the challenge of definitively diagnosing syphilis in tissue, and where the utility of having that molecular test becomes really important for patient care. And I'll note that the inflammation that was seen on the stomach, or seen in the stomach biopsy, was also seen in the colon biopsy, but the IHC did not pick up any spirochetes. So when the RPR came back positive, all signs pointed to syphilis, and possibly that had an accelerated clinical course due to the patient's untreated HIV. So he was treated for syphilis, specifically using neurosyphilis treatment with IV penicillin G, and his symptoms started to resolve after several days. When we got those stomach and colon biopsies back from Northwest BioTrust, the first thing that we showed was by qPCR was that, yes, there's indeed T. pallidum DNA in the colon, which squares with the inflammation that was seen in the biopsies. So we sequenced both the stomach and the colon and confirmed the co-infection by Nichols and SS14 strains in the stomach, but actually not in the colon, where Nichols really predominated. And I also asked Shaw Bakash, a research scientist in the lab, to develop a droplet PCR that could differentiate between the Nichols and SS14 lineages to give us more accurate ratios of what we saw, of what we were seeing. And so we found that there's about a 60-40 ratio of SS14 to Nichols in the stomach, but in the colon, it's 97% Nichols. And it just so happened that we had previously worked with Dr. Matt Golden's team at King County Public Health to sequence some remnant samples from Seattle, and those spanned exactly the period that the patient got sick. So the strains recovered from the patient are labeled with arrows there, and you can see how closely related they are and to other samples from, these are all samples from Seattle. And interestingly, they're interspersed with clusters of both heterosexual individuals in the SS14 and men who have sex with men in the nickels. And before I flip to the next slide, what I'd like you to focus on is in this heterosexual cluster. You see how the stomach sample diverges a little bit from the rest of those strains? I wanted to dig into that a little bit and see how the strain recovered from the stomach of the patient had differed from the rest of the strains in that cluster. And instead of a few random SNPs, which is what I was expecting, I found that the entire MRCA allele from the SS14 strain was being replaced by the Nichols sequence. And you can see that just at that locus, the proportion of SS14 to Nichols DNA was much lower. Now, this is a penicillin binding protein homolog, but no variants have ever been associated with penicillin resistance. So we don't really know what the biological significance of this mutation event is, or if there was a stimulus that caused it to happen. And while it was recognized that, it's been recognized that there are a few mutations in MRCA that tend to recur, which suggests some sort of selective pressure on it. The horizontal transfer and homologous recombination at this locus have never been observed before. And actually, this is kind of a big deal because this is the first time that T. pallidum has ever been caught in the act of recombination. So there's phylogenetic evidence of it having occurred in some membrane proteins, and you can see that there's a few instances where those have been transferred between strains, but it's never been seen happening in a patient sample. And I just want to highlight the last analysis that I did, which was looking for intrahost evolution of the Nichols clade strain by examining differences between the stomach and colon samples. And I found something really interesting in TP0865, which is an outer membrane protein. And so here you can see a cartoon of the protein domains. So all of the labeled features, including the hatch motif and these ECLs, are host-facing. In the stomach, we found two variants, as expected, and those were the same as the TP0865 from the most closely related strains circulating in Seattle. And I've colored the sequence features that allow us to discriminate between SS14 and Nichols sequences, and most of those ECLs are different by a few amino acids. In the colon, since remember, it was 97% Nichols clade DNA, I expected to only see sequences that looked like the Nichols allele from the stomach. But instead, I kept finding reads where Nichols and SS14 sequences were on the same amplicon, like this, which suggests another recombination event. And it seemed pretty heterogeneous. So this was really challenging to confirm. Remember that this is from really fragmented FFPE DNA, so we couldn't PCR the entire allele and just do long-read sequencing. That would have been great. Instead, Shaw designed these overlapping amplicons of about 400 bases, and we then sequenced those on the MiSeq. And those allowed us to get linkages between the hatch and ECL2, and then ECL2, 3, and 4. And I'm gonna spare you the details of exactly how we put those pieces together to infer the haplotypes. But we found multiple variants. Multiple different recombination events. So, for example, in variants 1A and B, ECLs 2 and 3 had been replaced, and 1B also had ECL4 replaced. Variants 2A and B also had picked up the SS14 hatch, but with a new G105D mutation. And then finally, we found low levels of these partial reversions to create novel chimeric ECL2 sequences. So this happened that they initially had the SS14 variant, and then there was another recombination event to take the Nichols sequences back as well. And so what does this actually mean for the organism or the function of TP0865? I don't know, but we have a few interesting ideas to chase. So let's first return to our original mission, which is vaccine design. And sequences derived from TP0865 are being investigated as vaccine targets. So let's see how widely distributed they are. These outer rings are colored corresponding to the sequences for the hatch, ECL2 and ECL4 from inside to outside. And ECL3 is a little bit trickier, so we're skipping it for now. And those red and blue represent the SS14 and Nichols variants that we found in the stomach for each of those genome features, the hatch, ECL2, and ECL4. And nowhere in this entire phylogeny of nearly 3,000 strains do we see that same mix and match organization of these particular sequences. They're totally novel alleles. And certainly immune pressure is one explanation for why the host-facing loops were swapped out in the colon TP0865 variant. But I think an equally important possibility is that this was an adaptive response to changing nutrient availability in the colon. Remember, our patient had lost 40 pounds and basically couldn't eat for months. And T. pallidum relies on its host for fatty acids. It's a fatty acid oxytroph. And I'm showing here the predicted structure of TP0865. It's a homologue of E. coli fetal, which is a fatty acid transporter. And as it happens, the Hatch and ECLs 2, 3, and 4, which are the ones that we saw changed in this patient sample, are the determinants of fatty acid substrate specificity. So ultimately, this is a cool case, but what did it tell us? Although there had previously been phylogenetic evidence that T. pallidum undergoes interstrain recombination, this is the first time that had been seen in a patient. And the fact that there were 2 genes affected was kind of not something that we had previously appreciated, and to me suggests that there's maybe a more robust mechanism for generating diversity than we previously understood. That said, we don't know why these 2 genes were affected, or if they're part of an evolutionary program to generate novel mutations. There's a lot more questions. In terms of medicine, I think some of the biggest open questions are around the role of the patient's untreated HIV and prior syphilis and how it affected the natural history of his infection. And obviously, there's a lot of things that we don't know in terms of the acquisition of the infections. And so there's a lot of questions there. And one thing I would like to note that even though it's pretty unlikely that these particular mutations from this patient are going to be transmitted from the stomach or the colon, in general, the more syphilis that there is, the more circulation, the more chance there is for co-infection and this interstrain recombination with unknown consequences. And overall, you can see how whole genome sequencing will help reveal the global diversity of vaccine targets. And this will, I think, be helped a lot by establishing rules and a central repository for process data. You can also see how sequencing on the worldwide level can give us kind of the contours of transmission patterns and also surveillance for the development of antimicrobial resistance. I haven't talked about the introduction of doxycycline and the use of doxypep, but one of the things that we're often asked is whether or not we're looking for characteristic mutations in the 16S. And yeah, any sample that comes across my desk, I'm going to continue to look to see if we see anything that looks like doxy resistance. So far, no. And then just the last thing is I hope I've demonstrated with TPRK and also the analysis of this co-infection case, how we're, I think, using some creative approaches to examine intrahost evolution and T. pallidum pathogenesis. And so I am just incredibly fortunate to work with many, many fantastic colleagues and collaborators. And I just have to say thanks to a lot of people, and most specifically Alex, who I didn't actually know any genomics when he hired me 5 years ago. I didn't know what to do with a FASTQ, and I also didn't know anything about viruses. But I think it's worked out pretty well, and I continue to have a lot of fun working on this project with a lot of great collaborators around the world. So I'm happy to take any questions.
That was fantastic. Thank you so much. Sorry, but Jeff.
I used to call it the spray. Since we know the genome, are there genes that somehow either participate in the uptake of DNA? You know, some bacteria are naturally competent with DNA. Are there genes that are recombinases that would allow for such?
Yeah, you know, it has Rec A, it has some of the binding proteins, it has the machinery to do homologous recombination, and it's used for, for TPRK. We don't know exactly what each gene does. We don't, we haven't been able to study them really, but it has the homologs. It does not seem to have a confidence system. We don't know how the DNA gets in. I'd love to figure that out. I don't know.
So Nicole, you mentioned vaccines a couple times. talk sort of in passing. I think the first mention was from a newspaper article from the 1950s. So where does that whole field stand today?
Still working. We are, it's really largely because of, we think because of TPRK, it's really hard to stay ahead of, to generate robust protection against it. But, Lorenzo is doing some incredible work in that field. And I think that we have a paper that was just recently submitted where we were able to show near complete protection following vaccination with a gene that I was also able to show that one of the particular epitopes in that in the vaccine antigen is conserved across all of the strains. So still working on it for sure.
The thing on line, I guess I have a question to build from that. So you have one candidate, it sounds like. Do you think that that with your genomic analysis, do you think that there's multiple? Do you think we're in sort of a good place to kind of have a multi-target approach, or are we kind of going to be really limited?
So I think that there are a lot of different approaches that could work. So I think that the team at UW is pursuing a few approaches that are more whole antigen. And there's a group at UConn that's doing kind of more of a mix and match approach where they're taking one ECL from this protein and another from over here and popping it onto a scaffolding protein and using that. I think that at this point, we're really just trying a lot of different techniques.
Josh, sorry.
Nicole, one of the things you highlighted was there are only a couple hundred samples that have been sequenced from the U.S. Like, what's your target? What's enough?
Well, I don't know that there's a number, but right now we have, I think, 14 states covered, and there are more than 14 states. So I think that before I put a number out there, I'd like to just see broader geographic coverage.
Is there any reason to think, or do you have any evidence that if you look at in individual heterogeneity of strains, is it more in samples from? Do you have any samples from congenital syphilis? Like, I'm thinking about immune pressure.
Oh, I would, yes. We don't have very many samples from congenital syphilis yet, but that is very much next steps. Yeah. Actually, to that point, I guess I'm putting in the Martin Reichenbach on a related topic. So we'll see.
Mark, what's different about the endemic strains? That's like, are there any clues from them that would allow us to understand the development?
Yeah, I'm sure there are lessons. I don't think we really understand. I don't think we really know what is all that different. There are certainly genes that are different, but I think it continues to be an open debate as to whether or not it is it not sexually transmitted because it's not sexually transmitted, or is it not capable of it in the case of Treponema, for example. So I don't know how much of that is due to the actual differences in the organism versus just the differences in how it is already circulating. And I know Sheila has thought a lot about this, so yeah.
Yeah, I would just say if you look at the Treponema in non-human primates in Africa, they infect non-human primates as well as they are sexually transmitted then.
Yeah.
And it goes to those animals.
Yeah. So
I think in humans it doesn't happen because of the timing. Most people that get yawns get it as children. They don't get it as sexually mature adults. They had it as children, which means it'd be less like.
Yeah. Right.
So I think it's time again more than biology.
Are you suggesting then that if sexually transmitted, they were exposed then as an older?
I would think so, yes. And some of the old data from the 50s on endemic syphilis indicate that there is likely sexual transmission of endemic and even congenital effects of endemic. So I think there's a lot more to this continuum than we know it.
Yeah.